Biotechnology is one of those fields that can seem impossibly modern. It brings to mind gene editing, DNA sequencing, custom cancer therapies, and the idea that medicine might one day be tailored to your exact biology. It sounds like the language of the 21st century, born in a world of supercomputers, molecular diagnostics, and laboratories filled with automated machines. But biotechnology is older than most people realize. Long before scientists learned to cut DNA, people were already using living systems to transform the world. They brewed beer, baked bread, made cheese, and fermented wine without knowing anything about genes, enzymes, or cells. They could harness biology long before they understood it. That is the deeper history of biotechnology. It begins with humanity learning to use life as a tool. Then it becomes a story about understanding heredity, decoding DNA, rewriting genes, and eventually trying to build a world in which medicine is not just for the average patient, but for the individual one. And that is what makes biotechnology such a powerful subject. It is not just the history of a scientific field, it is the history of a growing ability to intervene in life itself. For most of human history, biotechnology was practical rather than theoretical. Farmers selected plants with desirable traits. Shepherds bred animals for strength, milk, wool, or temperament. Brewers refined fermentation methods. None of this required molecular biology. It required observation, patience, and trial and error. In a basic sense, people were shaping living systems thousands of years before they could explain how inheritance worked. The modern story begins much later, in the 19th century, with a series of discoveries that transformed biology from craft into science. One of the most important figures was Gregor Mendel, the Augustinian monk whose experiments with pea plants revealed that traits are inherited according to predictable patterns. Mendel did not know about DNA. He did not know about chromosomes in the modern sense. But he showed that heredity had structure. It was not just a vague blending of parental traits. Something discrete was being passed on. That mattered enormously because it suggested that life followed rules. At around the same time, microbiology was being revolutionized by scientists like Louis Pasteur, who showed that microorganisms were responsible for fermentation and disease. That was another critical shift. Fermentation was no longer a mysterious transformation. It became a biological process that could be studied, controlled, and improved. These discoveries laid the groundwork for modern biotechnology. They showed that life was not just alive. It was organized, lawful, and therefore potentially engineerable. The 20th century pushed that idea much further. As genetics advanced, scientists began connecting inheritance to physical structures inside cells. Chromosomes became central to the story. Then came one of the biggest turning points in the history of science: the identification of DNA as the molecule that carries genetic information. Once DNA entered the center of biology, the field changed direction. In 1953, James Watson and Francis Crick, building on crucial experimental work by others, including Rosalind Franklin and Morris Wilkins, proposed the double helix structure of DNA. That discovery did more than explain what genes were made of. It suggested how biological information could be stored, copied, and transmitted. For biotechnology, this was like discovering the code behind life's operating system. If genes carried instructions, and if those instructions were written in a molecular language, then the next question was obvious. Could those instructions be read? Could they be copied? Could they be changed? In the decades that followed, biology became increasingly molecular. Scientists learned more about how DNA directs the production of proteins, how genes are switched on and off, and how mutations can alter function. The old biological world of visible traits and whole organisms was being joined by a new world of sequences, enzymes, and information. And then, biotechnology made a giant leap. In the 1970s, researchers developed recombinant DNA technology. This was the moment when scientists gained the ability to cut and recombine genetic material from different sources. For the first time, genes could be moved in deliberate ways. DNA was no longer just something to study. It became something to manipulate. This changed everything. Biotechnology was no longer simply about understanding life. It was about redesigning it at the molecular level. The early years of recombinant DNA were exciting, but also unsettling. If scientists could combine genes across species, what exactly were the limits? What counted as acceptable experimentation? Could engineered organisms escape the lab? Could biotechnology outpace society's ability to regulate it? Those concerns became serious enough that scientists themselves called for caution. The famous Asilomar Conference in 1975 brought researchers together to discuss the risks and rules of recombinant DNA research. That moment matters because it set a pattern that still exists today. Biotechnology has always advanced alongside ethical anxiety, and yet the scientific momentum was impossible to stop. Once recombinant DNA techniques existed, biotechnology became commercially important very quickly. One of the earliest major successes was the production of human insulin using genetically engineered bacteria. Before that, insulin for diabetics often came from animal sources. Recombinant insulin showed that engineered organisms could make medically valuable human proteins at scale. That was a landmark. It proved biotechnology could move from laboratory theory to practical medicine. It also helped launch the modern biotech industry. Startups, research firms, pharmaceutical companies, and investors began to see biology not just as science, but as a platform technology. If the 1970s were the birth of modern genetic engineering, the 1980s and 1990s were the period when biotechnology became industrial. New tools accelerated the field. One of the most important was polymerase chain reaction, or PCR. PCR made it possible to amplify tiny amounts of DNA into enough material to analyze in detail. That may sound technical, but its importance is hard to exaggerate. PCR made genetic analysis faster, cheaper, and far more practical. It became essential in research, diagnostics, forensics, and later in many forms of medical testing. At the same time, scientists got better at sequencing DNA, identifying disease-related genes, and building more precise ways to study cells and proteins. Biotechnology was no longer one technique. It was becoming an ecosystem of methods. Then came one of the grandest scientific projects of the modern era, the Human Genome Project. Launched in 1990, the project aimed to map and sequence the entire human genome. It was massive, expensive, and deeply ambitious. The idea was simple to state, but monumental in practice. If scientists could read the full genetic instruction set of a human being, they could understand disease more deeply, identify biological pathways, and build a new foundation for medicine. The Human Genome Project changed biotechnology in two ways. First, it accelerated the development of sequencing tools and computational biology. Second, it shifted the field's imagination. Biotechnology was no longer only about making one engineered product at a time. It was about information on a whole new scale. Genes, variants, expression patterns, biomarkers, and genomic databases became central to the future of medicine. This is where the story starts bending toward personalized medicine. Traditional medicine is built around populations. A drug is tested in large groups. A treatment guideline is written for the typical patient. A dosage is standardized. That model works remarkably well in many cases, but it also hides an obvious fact. Patients are not biologically identical. People vary in their genes, metabolism, immune response, disease risk, and treatment sensitivity. Biotechnology made it increasingly possible to measure those differences. At first, that meant genetic tests for specific inherited disorders. Later, it meant screening tumors for mutations, identifying biomarkers that predict response to treatment, and using genomic data to sort patients into more precise categories. Medicine began moving away from one-size-fits-all thinking. But before personalized medicine could fully take shape, biotechnology had to cross another threshold. It had to move from reading genes to editing them. Gene editing had earlier roots, and scientists had long explored ways to alter genetic material. But the field changed dramatically with the arrival of more practical editing tools. Zinc finger nucleases and talins were important steps because they showed genes could be targeted more deliberately. But these methods were technically demanding and difficult to scale. Then came CRISPR. CRISPR, adapted into a gene editing tool in the 2000s, transformed biotechnology because it made targeted editing faster, cheaper, and more accessible than earlier methods. Instead of treating the genome as something almost impossible to rewrite precisely, CRISPR made editing feel programmable. That was a conceptual revolution. For decades, biotechnology had mainly read, copied, and inserted. Now it could cut with far greater precision. That opened the door to correcting mutations, modifying cells, creating better disease models, engineering crops, and rethinking what treatment might mean in genetic disease. The excitement was immediate. So was the fear. Because gene editing raises some of the hardest questions in modern science. If you can correct a disease-causing mutation, many people would call that a medical triumph. But what if you move beyond treatment into enhancement? What if you edit embryos? What if changes can be inherited? What happens when the power to modify biology outruns public consensus about how it should be used? Those questions became impossible to ignore as gene editing improved. Biotechnology had entered a new phase, one in which the ability to intervene in life was no longer limited to observing genes or manufacturing proteins. It now involved rewriting the code itself. At the same time, another branch of biotechnology was reshaping medicine from a different angle: cell therapy and immunotherapy. Instead of giving patients only drugs made in factories, scientists began engineering living cells to become treatments. In some cancer therapies, immune cells can be modified to better recognize and attack tumors. This is one of the most striking examples of biotechnology's broader transformation. Medicine is no longer just chemistry. Increasingly, it is becoming biological design. This shift helps explain why personalized medicine has become such a powerful idea. Personalized medicine does not mean every patient gets a completely unique drug made from scratch. More often, it means treatment is guided by specific information about that patient: a genetic mutation, a biomarker, a tumor profile, a predicted drug response, or a molecular signature that places them in a more precise clinical category. In cancer care, this approach has already changed practice in important ways. Some therapies are prescribed only when a tumor carries a particular mutation. In other areas, pharmacogenomics helps doctors understand how a patient may metabolize a drug, which can influence dosing or treatment choice. Genetic testing can also reveal inherited risks, allowing earlier screening or prevention. That is one of the central achievements of biotechnology. It has helped medicine move from treating diseases as broad labels to treating them as biological mechanisms. And yet, the reality is more complicated than the promise. Personalized medicine is powerful, but it is not magic. Biology is messy. Many diseases involve multiple genes, environmental factors, lifestyle, timing, and chance. Even when genetic data is useful, it may not tell the whole story. A mutation might increase risk without guaranteeing illness. A targeted therapy might work brilliantly in one patient and poorly in another. Data can refine medicine, but it does not eliminate uncertainty. There are also social questions. Who gets access to these technologies? Who owns genetic data? How should privacy be protected when your genome contains information not just about you, but about your family? Will personalized medicine reduce inequality, or deepen it by concentrating advanced care in wealthy systems and populations? These are not side issues. They are part of the history of biotechnology itself. From recombinant DNA to gene editing, each major advance has been followed by a struggle over regulation, ethics, ownership, and trust. Biotechnology does not just change what is possible in the lab. It changes what societies have to decide about life, risk, and fairness. That is why the field's history matters so much. Biotechnology did not begin with CRISPR, and it did not begin with precision oncology. It began when humans first learned that living processes could be harnessed. It accelerated when heredity became measurable. It transformed when DNA became readable. It became an industry when genes became engineerable. And it entered a new age when the tools of biology became precise enough to diagnose, target, and sometimes even rewrite the mechanisms of disease. Seen that way, personalized medicine is not a sudden invention. It is the latest chapter in a very long story. A story about moving from observation to control, from breeding to genetics, from genetics to molecular biology, from molecular biology to engineering, and from engineering to medicine tailored, however imperfectly, to the person. That path helps explain why biotechnology feels so consequential. It sits at the intersection of information, industry, medicine, and identity. It asks what life is made of, how much of it can be changed, and who gets to decide. And in that sense, the history of biotechnology is not just about science. It is about power. The power to diagnose earlier. The power to manufacture therapies differently. The power to edit cells. The power to predict risk. The power to treat patients not as averages but as individuals. Whether that power ultimately becomes more humane, more unequal, or both at the same time may be one of the central questions of modern medicine. But the direction of the history is already clear. Biotechnology began with people using biology without understanding it. Then came the age of understanding. Then the age of manipulation. Now we are entering the age of precision. From gene editing to personalized medicine, biotechnology has been a long effort to move closer and closer to life's internal logic, and then to use that knowledge to change outcomes. That project is still unfinished, but it has already changed the meaning of medicine, the meaning of inheritance, and perhaps even the meaning of what it means to intervene in human life. And that is why the history of biotechnology is not just the story of a field. It is the story of humanity learning, step by step, how to read the code of life, alter it, and then try to heal with that knowledge.